I am Dr. Rupali Sharkar, Department of Physiology. I am honored that my department allowed me to take the responsibility to introduce our honorable speakers before the audience. Well, today we are presenting neurodegenerations in Alzheimer's disease in our technical session presented by Professor Dr. Tushar Kanti Ghosh. Dr. Ghosh is a professor, Department of Physiology, University College of Science and Technology, Kolkata. He did his master's and PhD in physiology from the University of Calcutta and postdoctorate from Harvard University, Washington, D.C., USA. A gentle, reticent human being, Professor Ghosh is happily free to travel the different arena of neurobehavioral physiology and electrophysiology. With this multifaceted background in neurophysiology, Dr. Ghosh published a lot of contributions in several national and international journals with high reviews like Brain Research, Journal of Neuroimmunology, Asian Journal of Farm Clinical Research, Journal of Immunotoxicology, American Journal of Physical Anthropology, and so on, along with guided students for their PhD degree too. Now, without further ado, we will turn the time over to Dr. Tusharkanti Ghosh, our first presenter today. I invite Professor Ghosh for his presentation. Professor Ghosh.
Okay, thank you very much. Thank you very much, Rupali, for your introduction, and good morning to everybody. At the outset, let me express my sincere gratitude to Dr. Pradeep Kumar Maiti, Principal of Ananda Mohan College, for inviting me in this webinar. I am also thankful to the organizer of this webinar, which includes my longtime friend Shonot, Rupali, Joyita, Arindam, Raghavendra, and many others in the department. Probably I know them very well. Ananda Mohan College was a part of City College once, and now it is separate. Now, when it almost 60, 70 years, it is separate college. City Group of College was started by a great educationist, Ananda Mohan Boshu, the first wrangler of Bengal, long time ago, in 1881. And that City College has grown in terms of time, and Ananda Mohan College is one of them. And today I'm really happy. I have long association with City College, that a part of City College has invited me to a webinar. I'm grateful to Ananda Mohan College, and I also remembered this great educationist. Probably we are here due to the movement created by those educationists in Bengal at that time. Now today, my topic is neurodegeneration in Alzheimer's disease. Alzheimer's disease, do you hear me? Alzheimer's disease is, of course, a neurodegenerative disease, and it is a disease of the associative function, most of the cognitive functions are deranged in that disease. So when the focal theme of this seminar is translational physiology, where I'm talking about a disease, which is pathology. In fact, these diseases are windows in the wall through which we can see many of the physiological processes. Brain is a very complex organ and it is very difficult to understand the mechanism of this complex organ of the universe. But as we see the sky through the window, probably these diseases are holes or windows to understand the mechanism of brain function, especially the associative functions of the brain. Now, lots of students are here, and I found that IUPC director, Dr. Somnath, Sandipan Sen, and Dr. Pradeep Kumar Maiti, principal, they are saying that they are not person of life science. You may be from English, you may be from chemistry, you may be from other branches, but brain is everywhere, and everybody know a little bit about the brain. So this is the cartoon of human brain, and we know there are three basic functions: motor, sensory, and associative functions. And more interestingly, these functions are organized from different regions of the brain. Say, for example, the motor function is organized from this area, sensory functions from this pink area. The associative functions are from these large areas. Therefore, the brain function depends on region. This is very important aspects of the neurophysiology. We call it a tonotopic organization or topographical organization. A particular region is related with a particular function. Now, the neurons and glial cells are the elementary components of brain. The brain is made up of neurons, basically neurons, and glial cells. The neurons, there are billions of neurons, each with an action and many dendrites. To stay healthy, neurons must communicate with each other, carry out metabolism, and repair themselves. So if this communication between the neurons are not accomplished, as you see over here, so these are the neurons, this is another neuron, this is a connection, synapse. So this is the basic neurobiology. So all the functions I mentioned, it may be motor, it may be sensory, it may be associative. They are dependent on this neural communication mechanism, the synaptic mechanism and neural circuitry. If this neural circuitry is affected, then the functions of the brain will be affected. In Alzheimer's disease, many of these functions are affected.
No.
Joyita, is there any problem? Neurodegenerative disease, there are different types of neurodegenerative disease. It may be Alzheimer's disease, it may be Parkinson's disease, Huntington's disease, amyotrophic lateral sclerosis, prion Kurzfeld-Jacobs disease. It is also called mad cow disease, transmissible spongiform encephalopathies. So there are different types of, these are the major neurodegenerative disease, and during this disease, some neurons are degenerated in a specific area of the brain. Say, motor neuron in basal ganglia may be related with Parkinson's disease. Neuronal circuitry in cognitive functions, mostly the associative area I told you previously. Whenever these associative areas are destroyed, then the Alzheimer's disease precipitates. There might be global CNS or ANS degeneration. That means whole brain may be degenerated, which can affect many functions. It may be sensory, it may be motor, it may be cognitive functions. Various cognitive functions are impaired in Alzheimer's disease due to neurodegeneration. These cognitive functions mostly the memory, emotional changes, maybe judgment, and also the autonomic changes. So Alzheimer's disease affects lots of cognitive function. It may be memory, it may be emotion, it may be abstract thinking, judgment, even the autonomic functions are also affected. I'll be talking about those specific changes later on. In Alzheimer's disease, as neurodegeneration occurs, this is normal brain, human brain. This is one photograph of normal human brain, and these are two photographs of brain from Alzheimer's disease. Look here, the changes. These are the gyrus, and this is sulcus. The gap between the gyruses. Here you see the gyruses are widened due to the destruction of the neurons in brain. So the whole brain is synced. As you see over here, the size is reduced. So this is a very severe disease affecting the whole brain. And it was first reported in 1906 by Alois Alzheimer, a German scientist, who reported a case study of woman in her late 40s who developed memory impairment and deficient in other mental faculties that worsened in severity until her death in her early 50s. So this is the first report by Alois Alzheimer. Later on, these type of patients are identified as Alzheimer patients. And today about 26 million people worldwide suffering from Alzheimer's disease. And demographic trends indicate the patients of Alzheimer's disease will increase, to 4 times by 250. So it is increasing enormously. So why it is increasing? So if you look at the demography, so this is from 216. So North America, Europe, Australia, so they are affected more. Africa less, India of course less, China in between, Russia in between. So lots of people are affected by Alzheimer's disease. In 2006, AD patients are 0.4% of the world population, about 26 million. And in 2050, the estimated prevalence of AD would triple, and absolute number of AD patients would quadruple. So you see, it will increase tremendous. Now, I like to mention a little bit about the neuropathology related with the disease. In preclinical condition, whenever there is no symptoms of Alzheimer's disease, but the pathological changes have already started. Signs of AD are first noticed in the entorhinal area of the cerebral cortex, this area. Do you see this area? And then it proceeds to hippocampus. Affected regions begin to shrink and the nerve cell die. Changes can begin 10 to 20 years before the symptoms appear. So whatever we see today, the molecular pathology, in fact, was started 10 to 20 years back. And the person may not perceive anything, because unless a threshold number of neurons are degenerated, the symptoms will not appear. So initial degeneration may not be noticed, because our brain is a unique organ which can compensate partial degeneration. So whenever the symptoms are coming, so there are lots of neurodegeneration. Memory loss is the first sign of it. So it is the preclinical one. The memory loss may be of different types. So any memory loss may not be linked with Alzheimer's disease. So after that, in Alzheimer's disease, the neurodegeneration spread to other areas, from entorhinal cortex, as you see over here, to the other associated area, prefrontal cortex and parietotemporal region. They are progressing, and the signs and symptoms are also increasing, which include memory loss, confusion, trouble handling money, poor judgment, mood changes, and increased anxiety. And whenever it is further increasing, the signs can include increased memory loss. So memory is gradually increasing. The memory loss is gradually increasing, and confusion, problems recognizing people, difficulty with language. You see, the language is also affected. Thoughts, restlessness, agitation, wandering, and repetitive statements. So these are the mild, moderate form, and whenever there is serious symptoms, as you see over here, the gaps here, between the gyrus, they are widened. Look at the ventricle. The ventricles are widened. So if you go to the previous one, you see they are not that much dilated. The gaps are less. In the preclinical condition, look here, they are not that much widened. So almost normal. But as the symptoms are increasing, the neurodegeneration is probably increasing and they are spreading to the other areas. As you see over here, in the severe condition, it has encompassed the cingulate gyrus, almost whole prefrontal cortex, parieto-occipital cortex, temporal area, and of course the hippocampus and entorhinal area. They are enlarged ventricle, and the symptoms include weight loss, seizures, skin infection, groaning, moaning, grunting, increased sleeping, loss of bladder. Death usually occurs from aspiration pneumonia or other infection. Caregivers caregivers can turn to a hosepipe for the help and palliative care. So these are the symptoms. Now, during this condition, the degeneration, I told you, they are almost affecting, in severe condition, they are affecting large areas of the brain, which includes the limbic system, association areas of cerebral cortex, basal forebrain, cholinergic system, striatum, thalamus, cerebellum. So large areas are, though initially it started with the association area, but later on it is affecting larger area, including the motor areas. Now, why the memories are affected more? Because the area, entorhinal cortex, hippocampus, prefrontal cortex, whatever I talked about, they are the passage of several important track, like mammalothalamic tract, vigdasa tract, and also the callosal fiber. So these tracts are affected, which ultimately reflected in the loss of memory. I don't want to discuss in the morning lots of neuroanatomy, which is boring sometimes. But even when there is no degeneration, the Alzheimer patient shows lower oxygen consumption. It can be assessed by PET. So whenever positron emission tomography scan was done on Alzheimer patient, it was found there is marked reduction of metabolism in posterior parietal cortex and some portion of the temporal lobe, because these regions, the neurons, as the neurons are degenerating, the oxygen consumption is decreased. As you see over here, this is the PET scan of normal brain, and this is from Alzheimer brain. As you see over here, the oxygen consumption is decreased because of the lower number of neuron over here. The causes of AD are not clearly understood, but the histopathological hallmarks of this disease are widely accepted. So whenever there is Alzheimer's disease, there might be different types of dementia. There are other neurodegenerative diseases where dementia may occur, but in Alzheimer's disease, two important characteristics are present. These are plaques and tangles. Plaques are extracellular deposit of a protein, we call it amyloid beta protein, which are surrounded by a shell of dystrophic neurites. Dystrophic neurites means they are abnormal dendrites of different types of neurons, and there are also substantial number of glial cells. So, and in the middle, there are some protein, they are amyloid beta protein, which is the focus of our discussion. There is also neurofibrillary triangle. They are intracellular, densely packed abnormal fibers. They occupy much of the cytoplasm, And as a result, the nucleus may be pushed out or may be destroyed totally. These fibers in many tangle-bearing neurons are so high in number that the nucleus is displaced and the cell body appears to be swollen. So these are the two important characters, plaques and tangles. They are present in the Alzheimer brain, especially in the affected areas. So if you look at the structure, so this is the AD plaque. As you see, there are lots of protein in the middle core and surrounded by, you see, you don't see it here, but there are also glial cells and the neurites of different neurons. And these are tangles. This is one neuron, lots of fibers. This is PHI, paired helical elements are there. So they are, in fact, nucleus is here. They are almost covering the cytoplasm. As a result, the cell may die. This is another picture from human brain. As you see here, these are the plaques. In the center, these are the amyloid proteins surrounding others. These are the neurons where the tangles are present. Now, the core of these plaques, they contain multiple species of amyloid beta protein. There are different species of amyloid beta protein. They are beta sit protein and carbohydrate moieties are there, but two important components are A beta 42, because 42 amino acid containing amyloid beta protein that is prone to aggregation is present there, and A beta 40 is also present there. See, these two proteins are found there. The tangles consist of pairs of twisted filaments with a highly regular periodicity. They are called paired helical filament, PHF. PHF are composed of cytoskeletal protein, the microtubule associated protein in tau. In the nerve cell, there are cytoskeletal protein. They are called microtubule. Microtubules are present all over the body in different types of cells, but in nerve cells, they are very important because they are associated with the transport of material from cell to terminal or terminal to cell. So they are very important in the transport mechanism, and these microtubules are formed by the tubulin protein, which are aggregated together by a stabilizing protein called tau. Whenever this tau is affected, the microtubules is disorganized, so the transport mechanism is affected, which leads to the degeneration of the neuron. So this is the amyloid precursor protein, or APP. APP, this is the protein. As you see over here, this is a very long protein, and this is the initial molecules for the formation of A beta protein, which is the cause of this disease. This protein is synthesized in the neuron but processed by two different pathways. One is called alpha secretase pathway. Alpha secrete is a protein. It can break down the APP into a large fragment, which is called large soluble fragment, or SAPP alpha, and C83 component by alpha secretase. Now this fragment SAPP alpha, that is involved in neuroprotection and synaptotrophic effect. So it is a physiological molecule. It is not a pathological molecule. It has physiological property. By synaptotrophic, they help in the formation of synapse and also protection of neurons. C83 is involved with gene expression. So this is a physiological process. This molecule is required for the brain growth, at least in the initial phase. But there is another pathway called beta secretase pathway. Beta secretase pathway, it is also a protease which break down this molecule into a part. We call it SAPP beta. It is not alpha. It is a different portion here. So this beta portion and the rest of the thing is called C99. C99 is further broken down by another enzyme called gamma secretase, and it produces two components. One is called amyloid beta protein. It may contain 38 to 43 amino acids, and AICT, APP intracellular domain. So these two proteins are coming, and A beta, as you see over here, this is the large APP. After breakdown of the beta site, we are getting this part. This is SAPP beta, and then C99. From C99, we are getting A beta, and these A beta proteins, they can aggregate. They are forming the core of the plaque, which initiates neurodegeneration. So in this cartoon, probably that can be visualized what I have discussed. So this is the protein APP embedded in the cell membrane. Two enzymes, they are coming, they are cutting the whole protein, and the components which are coming out, they are aggregating extracellularly, forming the core of the plaque, and where the neurites, glial cells are coming and forming the plaque. So why I'm talking about this material, because these A beta appears to be responsible for neurotoxic effect. Mechanism of neurotoxicity, if you look at in the Alzheimer's disease, A beta appears to be responsible. Soluble A beta. So this is aggregated A beta, you see what you see in the plaques, but there are soluble component of A beta. Whenever A beta is formed, normally, physiologically 10% of APP are processed by beta secretase pathway. So whatever A beta is formed, that is transported from brain to circulation through a special mechanism in the brain. But if the production of A beta is increased due to some reason, it may be genetic also, then this A beta aggregates. So before A beta aggregation, there might be soluble A beta oligomers, and several investigators have shown that the soluble one may be more toxic than this aggregated one, because the soluble A beta, they impair functions of synaptic ion and glucose transporter with loss of synaptic plasticity. Synaptic plasticity is related with memory. So whenever this plasticity is affected, memory is impaired, and that's why it is the first sign of Alzheimer's disease. Whenever there is even no neurodegeneration, but there is functional abnormality due to the soluble A beta effect on the synaptic function. A beta oligomers are associated with microglia activation and astrocytosis that initiated inflammatory response. A beta not only affect the synapse, but microglial cells which are present in the brain. There are 10 times more than, the number of neurons which are present, 10 times more cells are glial cells. There are different varieties of glial cells. Microglial cells are activated by A beta 42, and as a result, they become inflammatory. These microglia are very unique because they are protective as well as they are destructive. So they are just like double-edged sword. They are protective if they are in certain phases, but whenever they are activated, they will destroy the neuron. And cytoskeletal abnormalities which result, that may also be initiated by A beta deposition. I'll be talking about that. So this is the mechanism of neurotoxicity by amyloid protein. This amyloid protein is the cause of this disease. And this is called central amyloid cascade hypothesis. It was formulated long time ago, and which shows that this A beta is originating from certain mutation in gene. They are called APP gene. APP genes, I told you, amyloid precursor protein. There may be POS1, PS2 genes. They are mostly component, these proteins are component of beta and gamma secretase path. So if they are abnormal, then there is increased A beta 42 production. So whenever they are produced more, they oligomerize and deposited. And whenever they are deposited, they affect the synapse. And not only synapse, they affect the microglia and astrocyte. As a result, they secrete more cytokines. And the synaptic effect and the cytokines, they cause synaptic and neuritic injury. Due to the injury, the neuronal ionic homeostasis, as I told you, the glucose transport and ion transport are affected. So the neurons, are not able to maintain their normal functions. Lots of kinase and phosphatase are altered, and these kinase and phosphatase, they may help in the formation of tangles. I'll be talking about that aspect later. And the tangles and those microglial activation, inflammation, they are causing neuronal and neuritic dysfunction, degeneration. As a result, dementia occurs. So world was very simple up to this point, and this one I told you that tau, this is called tau pathway. I told you about the microtubules and the tau protein. Whenever tau protein are hyperphosphorylated, not only hyperphosphorylated, if there is glycation, ubiquitination, oxidation, truncation, then they are disorganized, and that may be initiated by glycogen synthase kinase 3 and cyclidine dependent kinase, PKA, PKC, etc. So, they are, whenever they are hyperphosphorylated, or rather the ratio between different isomers and degree of phosphorylation, then these tau proteins, they are dissociated from the microtubules, and as a result, these are the neurons. In the neurons, these are action, these are the microtubules stabilized by these small tau proteins. Whenever they are aggregated, the microtubules are disorganized. So transport mechanism from cell to periphery, periphery to cell, they are affected. So the neuron are not able to survive. But this central amyloid hypothesis, it has problem. Progressive dementia cannot be correlated with plaque numbers. So plaque numbers are not increasing with the progression of disease. Suspected AD pathology, they are called SNAP, lacking A beta deposition. Remember here, in some certain patients, they are called SNAP, SNAP, they don't have beta deposition. But neuronal injury they have, which are converted to mild cognitive impairment in 10% cases within 1 year. So there are certain patients where the A beta may not be present. The neuronal injury can at least partly occur independent of A beta related process. Neuronal injury can occur in independent, if so long I am talking about A beta is the disease, now I am saying some information where A beta may not be the root cause. So drug developed on the basis of those findings, they are not effective in all patients. So what is the situation? Situation is a little bit complex. So in familial type of Alzheimer's disease, whatever I discussed, this amyloid cascade hypothesis is probably operating. But they are only 2 to 5% of the AD patients. In familial type, this is due to the genetic mutation in chromosome 21, which is APPG, presenilin 1, presenilin 2 gene, 14, and 1 chromosome they are present, they are autosomal dominant. So the pathological mechanism I discussed so long, that depends on the mutation of this gene, but 95% of the patients are sporadic. We don't know the mechanism operating over there. But vascular lesion, brain injury, diabetes, air pollution, disrupted homeostasis of biometals such as copper, iron, zinc, infection, gum infections, spirochetes, even the flu infection. In 1918, the Spanish flu was followed by a neurodegenerative disease in Europe, which includes Parkinson's disease and dementia. And we do not know whether the pandemic now prevailing in the world will also be followed by another pandemic in neurodegeneration. Neurodegenerative patient may increase for the next few years after this pandemic. So neurobiologists, neurophysiologists are thinking that the changes which occurred in 1918 may also here, but parallel changes is always not true. However, the sporadic changes may be affected by certain gene apolipoprotein 4 allele or term M2 gene. These genes can increase the susceptibility of sporadic disease. So now I am coming to sporadic animal. In the sporadic Alzheimer disease. So in mechanism of Alzheimer disease, we talked about A beta 42, but there are also other mechanisms. These mechanisms include the neuroinflammation, production of ROS, membrane lipid peroxidation, generation of neurotoxin 4-hydroxy nonenal. So whenever they increase, they can cause degeneration. Release of glutamate from glial cells. Glial cells are present mostly the astrocytes in the brain. I told you they are 10 times more than neuron. They can release glutamate in certain situation, and this glutamate, whenever they stimulate NMDA receptor, that can initiate calcium entry within the cell neurons, and that can destroy. This is called calcium exocytosis. And A beta accumulation of NMDA glutamate receptor, whenever A beta binds with NMDA receptor, that can also initiate the degeneration process because they increase intracellular calcium, which activates a cascade involving neuronal nitric oxide synthase. This nitric oxide synthase, whenever activated, they are generating lots of and nitric oxide increase oxidative and nitrosative stress and mitochondrial injury. So there are other mechanisms. It is not that much simple. So AD is a disease with single sequential pathological path. AD as a disease with single sequential pathological pathway with A beta considered as the only initial causal event is likely to be progressively replaced by more complex picture in which AD is considered as a multi-parametric pathology that is subtended by several partly independent pathological process. So AD is not simple central amyloid hypothesis. So there are other mechanisms. This is a comment from the Nature Review 2013. So scientists, neurobiologists always try to find out what are the mechanism of neurodegeneration. There are lots of ways. One of the ways is experimental animal. The transgenic animal, they can give clue to the neurodegeneration in the familial patients, but for sporadic patients, some chemically induced animals are used. They are colchicine-induced animal model. There might be celecoxib or streptozotocin, the colchicine-induced model. Aluminum, they also help in the triangle formation. Celecoxib, streptozotocin may be injected. Beta amyloid, that is also related with the central amyloid hypothesis. Pesticides-induced AD, they are more or less very close to sporadic patients. So chemically induced animal model, they are used to understand the mechanism of neurodegeneration in sporadic AD. And in the next few slides, I'll be talking about the results from my laboratory. So we can inject stereotaxically to different inject these chemicals to different regions or even in the CSF, either in the hippocampus or in the CSF. We can inject colchicine, a plant product, which destroy those microtubules in certain amounts, and as a result, the animals will show certain behavioral and pathological characteristics of Alzheimer's disease. Similarly, streptozotocin may also be injected, but time will not permit to discuss all the things. So I'll be concentrating on colchicine injected AD rats. So after injection, we looked at memory in radial arm-image. So in animal, the memory parameters can be measured by radial arm-image. So there are different parameters I am presenting here: working memory and the reference memory error. So these are increased in colchicine animal. Neurodegeneration that occur in the brain can be assessed by nasal granule staining or Congo red staining of the plaques. Nasal granules, they will stain the nasal. Whenever these nasals are decreased, they indicate the neurodegeneration, a loss of functions of the neuron. Congo red indicates the presence of the plaque. And inflammatory markers in the hippocampus, we looked at hippocampus, initially looked at different portions of the brain, but as the changes are maximum in the hippocampus, so I am presenting here the data on ROS, reactive oxygen species, nitrite, TNF alpha, IL-1 beta, COX-2, prostaglandin. So these parameters are measured in an attempt to identify how the needed neurodegeneration. So we looked at behavior, the memory is affected, we looked at pathology, neurodegeneration occurred, we looked at the inflammatory markers and tried to find out how the behavioral aspects are related with the inflammation. So what we did, we used different doses of COX-2 inhibitor, heterocoxib, and whenever the neuroinflammation is affected, how they are reflected on the behavior. Look here. So this is the working memory impairment. So these are the normal animals. Whenever colchicine was injected, the impairment, as you see, the working memory errors were increased. But whenever we are giving the heterocoxib, they are coming back almost to the normal. So here you see heterocoxib, of course, there are remarkable change. As you see over here, the memory impairment may be blocked by heterocoxib, suggesting the neurodegeneration is probably inhibited by heterocoxib. Reference memory, same thing as you see over here, the degeneration, and they are coming back to the normal. And if you look at histopathology, this is 15-day study. So after injection, we look at 15 days. This is control. SAM, AD, look at AD, the nasal granules are less. In 21 days in AD, they are further lesser. Whenever I am using heterocoxib, 10 milligram, 20 milligram, 30 milligram, as you see over here, the granules are reappearing. That means it can not only affect the behavior, but histopathology. If you use the heterocoxib, which blocks COX-2, the neurodegeneration are less. And at 10 milligram, they are in fact not that much prominent. At 30 milligram, they are much more prominent, suggesting it is a dose-dependent effect and it can affect the neurodegeneration. Here you see quantitative measurement of nasal granules. So this is 21-day study. This is 15-day study with different dose of heterocoxib, 10, 20, 30. You see the nasal granules, they are in fact increased. I mean the nasal granules concentration, they are increased. If you look at the Congo red, in 15-day study, few, in AD animal, few, these are the plaques. In 21 days, more plaques. Now, whenever I am using 10, 20, and 30 in AD animal, here you see the plaques are disappearing. Here you see the plaques at higher dose, they are not presenting, suggesting neurodegeneration is probably inhibited by heterocoxib. This is another. Now we look at the hippocampal ROS level. This is 21-day study. This is 15-day study. Now, whenever we are using different doses, as you see, they are coming down. These are the normal ones. So ROS levels are going down by the heterocoxib. Nitrite level, which indicates the nitrosative stress, they are also decreased by heterocoxib. TNF alpha level, this is pro-inflammatory cytokines. So these pro-inflammatory cytokines, now you can see even in the newspaper that there is cytokine storm due to COVID, and TNF alpha is one pro-inflammatory cytokine. So there is also a storm in the brain due to this inflammation, and they are coming back to the normal level after heterocoxib injection. 1 beta, this is also one inflammatory, and you see they are also going down to the normal level. The COX-2, we also measure the COX-2 level, the enzyme, and this enzyme, as you see over here, tremendously high. In 21 and 15 days, they are coming back to the normal level. Now, prostaglandin, it is also related with COX. This prostaglandin causes inflammation. They are also reduced in the hippocampus. So all these information suggest colchicine induces inflammation in hippocampus and causes memory impairments and neurodegeneration. Heterocoxy, a COX-2 blocker, can inhibit neuroinflammation and can block colchicine-induced neurodegeneration and memory impairment. So in this model, we found that COX-2 is playing important role for neurodegeneration, which is reflected on memories and neuropathology. Now, we also use NOS blocker. NOS blocker aminoguanidine was used to assess the role of NO in AD rats. Similarly, we look at those behavior, working memory, reference memory. So this is the normal behavior. They are impaired. Both of these behaviors are impaired in CAD rat, but after aminoguanidine, you see they are coming back with the different doses, of course. They are coming back to the normal, but they are not that much prominent like that of heterocoxy. We looked at the plaques, the number of plaques. These are the photographs, but look here, the number of plaques, they are increasing whenever we are using 30, 50 milligram per kg of aminoguanidine. This is the without, that means this is CAD rat. Their plaques are, numbers are decreasing. So if you look at the ROS level, this is AD rat, ROS level is decreased with the 2 doses of aminoguanidine, and also the nitrite level is also decreased in hippocampus. So we look at TNF alpha and 1 beta. This is TNF alpha. TNF alpha level is very high, as we see in the previous slides, and they are coming very low at low level. And also IL-1 beta level is low. This effect is more pronounced compared to the heterocoxy. So aminoguanidine, a NOS blocker, can also inhibit neuroinflammation in CAD rat. So NOS is also important, not only COX. Now vitamin C, an antioxidant, was used to assess the role of neuroinflammation in memory impairment in CAD rat. We also use vitamin C. So why we use vitamin C? Because vitamin C is antioxidant, and here you see again the memory impairment occur, but whenever I am using vitamin C in 3 different doses, their impairments are decreased. But it is not remarkable like heterocoxic. Whenever you look at the nasal granules, these are the nasal granules CAD rat, 3 doses are used, 200, 400 and 600. You see in 200 and 400, the nasal granules are regained, but whenever 600s are used, they are further decreased. So there is biphasic effect, not like aminoguanidine or heterocoxy. Vitamin C is probably acting as pro-oxidant and antioxidant. So at lower dose, it is probably antioxidant. As you see over here, the plaque numbers are decreased at 200, 400, but it is increased at 600. So suggesting TNF alpha, they are showing similar type of response. TNF alpha and also IL-1 beta. So at higher dose, they are increased, suggesting, so ROS, I don't want to bore you all those data. Vitamin C has a dual effect on colchicine-induced neuroinflammation in CAD rats. So dual effect, pro-oxidant and antioxidant at lower dose, pro-oxidant at higher dose. So vitamin C may not be always good. Now NMDA receptor, I told you, that is the receptor for glutamate, and that can also participate in the neuroinflammation. So we use memantine, a drug which blocks the NMDA receptor. And here we see whenever we are using memantine in 14-day study and 21-day study, the ROS concentration, as you see over here, they decrease from the affected animal 140 to 130. Similarly, in 21-day, it is more pronounced. And also, if you look at those data in nitrite, they are also remarkably decreased by NMDA receptor blocker. If you look at TNF alpha, IL-1 beta, look here, TNF alpha, they are also decreased. In 21-day study, it is more pronounced. Same thing in IL-1 beta. If you look at the nasal granules, same story. The memantine can cause a prevention of the neurodegeneration. So summary, inflammatory change are found in CAD rat and in hippocampus, along with memory impairment and neurodegeneration. Inhibition of neuroinflammation in CAD by heterocoxy, aminoguanine, and vitamin C also reduces memory impairments and neurodegeneration in those animals. So, if we talk about this whole experiment, the possible mechanism are there. Whenever we are injecting colchicine in CSF, there is microtubular disruption, which blocks exoplasmic flow, increased ROS and NO generation, increased cytoskeletal production. So whenever you are using vitamin C or aminoguanine, they are affecting these ROS or NO. And if they are not inhibited, they are activating COX-2 enzyme. So COX-2 enzyme increase production of prostaglandin, and this prostaglandin activate microglia. They are further help in the production of neuroinflammatory cytokine, IL-1 beta, IL-6, TNF alpha. They are causing neurotoxic effect, neuronal death, and memory impairment. Or they also act through efflux through blood-brain barrier change, peripheral immune cell changes. Whenever we are using heterocoxy, they can block these COX-2. So by using vitamin C, aminoguanine, heterocoxy, we can block this process and the neurodegeneration. So on the basis of this, drug may be developed, but as I have mentioned, a drug may not be applicable to all the AD patients. In fact, AD is incurable. Though there are drugs, it may be as anti-acetylcholine, antioxidant, as I told about vitamin C is also used, folic acid, cholesterol lowering drugs are also used, anti-inflammatory drugs. I told you they are not always very effective in all the patients. Immunization with A beta in transgenic mouse model, they have little bit of effect. Now there might be secretase inhibitor. I don't know my time. A beta fibrillation, anti-tau drugs, even the estrogen was found to be effective in some patients. Now management of AD, I told you about medication, psychological intervention, caregiving, these are the most important thing. Now for prevention, so whenever you don't have cure, you must look for prevention. So diets good, which prevent this disease. The diet rich in flavonoid, tea, coca, red wine, vitamin E, C, folic acid, omega-3 fatty acids, the food which contains those, they're good, they prevent this disease. Diet to be avoided, high sugar, high saturated fat. So whatever you take as the junk food, fast food, and high simple carbohydrates. So don't drink those Coke or Pepsi. And prevention in lifestyle. Lifestyle is most important. AD reduces inactive intellectual. If your intellectual activities are increased, then that will be good for brain function. Intellectual activity, reading, crossword puzzle, language learning. If you learn language, play board games, social interaction, physical activity, they will inhibit neurodegeneration. So our lifestyle, if you look at the lifestyle, so human cravings are there, drive for money and power. So whenever there is drive for that, this craving is probably affecting not only your physiological system but brain. So they are affecting the brain more. But if you expand the scope of conscious mind, if you use your conscious mind, if you use the intellectual activity, creative mind, probably that will inhibit this degeneration process. This is the paintings of Jomo, a Tibetan painting, 17th century. It was collected from Tibet, the Potala Palace. So the work I presented here, I am thankful to UGC, DST, NTRF, DRDO for their financial assistance. The work I presented here mostly done by Sushmita Sil. She is now instructor in Nebraska University, Omaha. Ruksha, Pritha, Orijit, they also help in this work. And at the end, I would say that these poor people who are mostly old, they need help. There is no cure for this disease, but the caregiving may be given. Thank you all.
Thank you very much, sir, for your wonderful speech about Alzheimer's disease, its development, and its mechanism of action, medication, and prevention. Uh, our students will be benefited a lot from your lecture, and there are some questions in the chat box. I am requesting Dr. Raghavendra Misra to present the questions to sir.
I told you that there are several genes, several genes which are related with Alzheimer's disease. In familial Alzheimer's disease, APP gene, presenilin 1 gene and presenilin 2 genes, they are present in certain family and they are autosomal dominant. So they reappear in those families. Those families are investigated in Europe and North America. So they are probably related with the disease. As I told you, whenever these genes are defective, they increase the formation of A beta 40, which result in this disease. But for sporadic one, the disease is mostly dependent on the environmental factors. Gene is not that much important. However, there are ApoE gene that can increase the incidence of the disease. Whenever a person have multiple copies of these genes, probability of this disease may increase in sporadic form. So these are the genetic implications of these two types of disease, familial and sporadic. Several. Okay, as I told you, the stress. In stress, there are different physiological changes, which include higher level of HPA activity, hypothalamo-pituitary adrenal activity, which affects the immune system. This immune system, I didn't discuss in this lecture, but this peripheral immune system, they can influence the brain activity. There are lots of receptors in the neuronal cell which are affected by these stress markers. So stress not only affects the peripheral system, but the stress marker can pass through blood-brain barrier, and that can affect the neuron, and there are receptors in the neuron which can interact with those stress markers. And these stress markers, so they are mostly cytokines. So cytokines I'm talking about, this TNF alpha, the IL-1 beta, so they are neurotoxic. So whenever they are able to enter in higher amount in the brain, they can initiate the neurotoxic mechanism. So stress may initiate the neurodegeneration process, but stress, in addition to the physiological mechanisms, stress can also be managed. As I discussed previously, stress can be managed by your lifestyle. So if you are able to withstand stress, it will protect you. And to maintain the stress, it is better to engage in cognitive dense activities like reading. Whenever you are watching TV, your brain is not that much activated. But whenever you are reading, you are thinking, you are writing, probably that will stimulate the synaptic mechanism, and that will prevent the stress-induced effect on the neuronal circuit. A beta, whenever it is increased, as I told you, A beta, whenever it is deposited in the plaque, it activates the microglia and probably astrocytes. So they are generating cytokines. And these cytokines, in turn, can affect the glial cells. They also affect the neurons. They increase the COX activity. Prostaglandin is coming into picture, so that initiate the production of more cytokines. These cytokines are neurotoxic, means they can influence the mitochondrial mechanism, and A beta, whenever it is soluble, probably Arindam is talking about that. It was found whenever A beta is soluble, it is not precipitated in the plaque, it is soluble, it can pass through the blood circulation. Whenever it is going to neuron and entering within the cell, A beta can directly influence the mitochondria. And mitochondrial destruction, maybe it is toxic in several ways, but the level of toxicity depends on its location. So soluble one is more toxic because it can enter within the cell probably and can influence the mitochondrial activity. A beta plaques are present outside the cell, but the soluble one can get inside, and that can influence lots of metabolic process, including the mitochondrial oxidative mechanism. So probably that initiates the neurodegeneration, and that's why A beta 42 is considered more toxic than the plaques. Now, cholinesterase, I didn't talk everything over here because in one lecture you cannot cover everything. There are many hypotheses. I talked about central amyloid hypothesis and to some extent the inflammatory mechanism. But there is another hypothesis, acetylcholine hypothesis. In the base of the brain, there is one nucleus, basal nucleus. It is cholinergic. It has lots of projections in the prefrontal brain and other regions. It was found in the Alzheimer's disease that these cholinergic neurons are degenerated. So initially it was thought this basal nucleus of minor that is causing this disease, and for that reason, and even today, anti-acetylcholinesterase drugs are used. So whenever these drugs are used, they can increase acetylcholine concentration in the brain, and that helps in the cognitive recovery. So in patients, the problem is cognitive impairment, memory loss. So initially, whenever these cholinesterase inhibitors are used, even those are used by the doctors, clinician today, the cognitive impairment is better. So cholinesterase enzyme, as I understand from the question, the cholinesterase can increase acetylcholine, and acetylcholine, whenever increased in specific neuronal pathway originating from the basal nucleus of brain, their functions probably are better, and that's why the cognitive loss is less. So this is the relation between acetylcholine and cognitive impairment. So I mentioned, I didn't know that the audience is more interested about those aspects. I have shown some diets and exercise. Diets rich in antioxidant, so containing flavonoid. So all the vegetables, green and leafy vegetables, they are rich in flavonoids. So whenever those flavonoids are present, they are in fact antioxidant. Probably you have noticed the number of patients of Alzheimer's disease is more in North America, Europe, Australia, but less in India, Africa, Europe. It may be genetic one, but the others are saying the food habit is most important. In India, lots of antioxidants are used by the common people, not only in form of green vegetable containing flavonoids rich in antioxidants, but lots of spices used in India, which include curcumin and coriander and other species. They are rich in antioxidants. And in one of my study, I didn't mention it here, we have used in another model, that is streptozotocin model, we have used that Zafran used in biryani. And we found the Zafran is able to inhibit neurodegeneration. So antioxidant rich food, those are fresh food, whole food. Don't take processed food. So those are good for neurodegeneration, for preventing neurodegeneration. And also the food rich in vitamin C, vitamin A, zinc. So whenever a food is rich in those components, they are better. And if you talk about exercise, I told you, exercise, if you talk about physical exercise, that will increase the muscle volume. So if you go to gymnasium, your muscle volume may increase. There might be 6 packs or 8 packs. But if you want to exercise the brain, then the board game, the language learning, or even you may write poetry or other creative events, that can exercise your brain. So the neural circuitry, if you use those neural circuitry by watching TV, you are not activating your neural circuitry. Neural circuitry will be activated more whenever the creative functions are performed by those neural areas. So whenever I mean exercise, physical exercise may also be good. Physical exercise also, they send information to the brain. But mental exercise, creative activities, probably they are better for preventing neurodegenerative. This is like Alzheimer's. Okay.
Thank you. Thank you very much, sir. Once again, we have more questions, but we will discuss this later. Now we are moving to the next part of our session. Thank you, sir.
Thank you very much. Thank you.
Our next speaker is Dr. Professor Birendranath Mollik. He is the professor of Department of Neurobiology and J.C. Bose National Fellow, School of Life Science, Jawaharlal Nehru University, New Delhi. I would like to request again Madam Dr. Rupali Sarkar to introduce Professor Mollik. Please, Madam.
Thank you, Joyita. Okay. And here comes out our second presentations, which is based on REM sleep. The topic is REM sleep is small investment, reaping large benefit, an efficient physiological process and evolutions. The speaker is eminent scientist, Professor Birendranath Mollik. Dr. Mollik is so familiar for his scientific work that he needs no introductions. But as it is our formal practice of technical session, I have to say some few words about him. Dr. Mollik is an Indian neurobiologist, senior professor of neurobiology, and J.C. Bose National Fellow at the School of Life Science of Jawaharlal Nehru University, New Delhi. He is well known for his research work on neurophysiological studies on sleep and wakefulness. Dr. Mollik did his graduations and masters in physiology from the University of Calcutta and moved to All India Institute of Medical Science, Delhi, for his doctoral studies as a teaching fellow. He joined the JNU in 1986 and an assistant professor, and between 87 to 2002, he visited University of California, Harvard Medical School, and University of Nice, Sofia Antipolis, USA, as visiting professor. His work has been cited in the number of publications, of course, both in national and international films. He has authored and edited articles and text on the subject, which included the first monograph on REM sleep, that is, rapid eye movement sleep. The CSIR awarded him country's one of the highest Indian Science Award, Shanti Sharup Bhatnagar Prize, in 2001 for his contributions in medical sciences, and become an elected fellow of all three major science academics: National Academy of Science India, Indian Academy of Sciences, and Indian National Science Academy. BK Anand Award, Shakuntala Amit Chand Award, ISMR, and National Bioscience Award, DBT, are the three feathers in the crown of Dr. Malik. Well, now I hand over this platform to Professor B.N. Malik for his presentation. Professor Malik. Hello, Professor Malik.
Yes, can you hear me all? Am I audible?
Yeah.
Okay, thanks to the organizer, the principal, Dr. Maiti, Rupali, Joyita, and rest all organizer for inviting me for this lecture. I'm grateful. And I guess this is a unique time that we are facing all over the world as we are heading or we are listening on through email, through the video conferencing. Okay, so can you see my slide now? Okay, so can you see my slides? Is it visible to you?
No.
No, sir. No, sir.
Okay, so now you can see me, right? Not the slides.
Yeah.
Full screen, and then can you see the slide now?
No, sir. Sir, you have to present once again.
Can you see now? Can you see the slide now? Okay, sorry for the delay. Whatever. Is it all right? Okay. So it is now 11:30 and I have this 40 minutes. So formally again, thanks for inviting me and good morning to all of you. See, as Rupali has so kind of Rupali to introduce with all these big words, nice words, thanks to her. As all of you know by now that I work on REM sleep mechanism, and this is my School of Life Sciences where the work has been carried out. Some of you might have visited sometime my lab. And I do work on rapid eye movement sleep. Can you see the cursor I'm moving? Can anybody say, can you see the cursor? Okay, the cursor as well, right? Fine. So REMS stands for rapid eye movement sleep. See, what I will do in my time allotted here, what I would say that this is the stage where all of us spend the least amount of time. So why evolutionary? We had to, or the nature has created it to maintain. And even with the least time one spends, it reaps such a huge benefit. I hope to be able to convey this to all of you in my next 40 minutes presentation. So evolutionary, what we see that there is movement and rest activity. Here I have shown some zebrafish, which here you can see immobile, the fish gets immobile. And here you can see even in ants, means I am showing at the lower species, be it vertebrate or invertebrate, all recorded in the labs. Now, so there is always basic rest and activity cycle, which is known as BRAC. And it is thought of the sleep and waking are modified form of this BRAC. This is some common visible sign that many of us must have seen. But then do we say that is sleeping or awake or somebody's dead or something? Like here also you can see. And I have shown from fish to human to dog to tiger to lion to hippocampus. Now the question, can anybody say what are these? See, wake or sleep or any state of mind is known from evolution. But then what we, the scientists, try to do is to have an objective way to classify and analyze. The picture that you are seeing here, what you would difficulty to understand because we do not have an objective way to understand. Here is a tiger. Now, if somebody tells me to go and touch the tiger, I would be reluctant because I don't know whether the animal is asleep or dead or waking or anesthetized. Therefore, it was necessary to identify different states of mind, which could only be done in late 1920s when the electrical signaling of the brain could be recorded. And that was EEG. Based on that, one could identify wake and sleep states. So from about late 1920s to about 1950s, here is Aserinsky and his daughter, who identified in humans and the babies that eyes are rolling over when somebody is asleep. And subsequently, Dewey and others, even William, what is his name? Stanford, Dement, they identified even in different animals, the eyes are moving even in animals during sleep. So they talked about, particularly Aserinsky, who named this state as rapid eye movement during sleep. So the sleep-waking were identified as active or quiet sleep or wake, and sleep, slow and deep sleep, and then REM sleep. This is a record in the cat, which is in my lab, but not that we are the inventor. It is recorded brainwaves, EEG, EOG, eye movement, muscle movement. And if you go through, you will find the EEG, the first pink, magenta or this brown colored waves, which goes into higher voltage and lower frequencies. But after that, suddenly it switches to like desynchronized, means higher frequencies, like we see during wake. But then eye movement starts, which is not there in deep sleep. In contrast, the muscle movement, which was very high during awake, significantly reduced during sleep, that further goes down almost atonic. This was identified be it in animals, as it is shown in the cat, and as it is shown in the rat, and of course in humans. This stage was identified as rapid eye movement sleep, and I will be talking about this stage. This stage is there in different animals, as it is shown here, different number of hours one spends, very high to low. This has a cyclic in nature. This is of course in human. The black bars are the cyclic that you are seeing. The more deeper you go into sleep, that increases. This is there in ontogenically, as you can see my arrow. That means even from birth to old age, though it is reduced, but never it is ending. And next slide I have done from different, a slide has been made that how in different species, be it from insect to invertebrate to fish, amphibian, reptile, birds, mammals within land and aquatic, how there are different stages of wakefulness or sleep, and then within sleep you have non-REM and REM. But in insect and invertebrate and fish, we do not have REM as such, because so far, as I showed you, that REM is rapid eye movement sleep, where the EEG brain, EEG desynchrony and all. So if there is brain not well developed, it is difficult to identify. Though we can identify still sleep and waking. So we do not know whether it is there in the very low species or not. But so far, even in higher species, opossum, ferret, cat, dog, human, elephant, zebra, people have identified these stages. And one is spending the least amount of time in this stage. Now, even if one has spent least amount of time, it is so important that if you talk about REM sleep is reduced in most of these disorders, and in some of these disorders it does increase. And experimentally, if you create REM sleep loss, there is irritability, aggressiveness, confusion, all these are increasing. Brain maturation, memory consolidation, they are decreasing. So this was the state of affair about mid-80s when even I started. So the question was, what does REM sleep, therefore, evolutionary, is there any necessity? Since it does all this functional and fundamental job, I thought REM sleep is likely to serve a fundamental function. It cannot be of vestigial significance. So REM sleep loss, there is a behavioral disorder, and since, as you can see here, most of these are related to brain, irritability, aggressiveness, confusion, anxiety, brain maturation, memory consolidation, I thought, what should be the difference between the brain neural tissue and the somatic tissue? One of the major differences is here you have excitability. So the REM sleep might affect this excitability, and if that excitability has to be affected, how do we study it? If excitability has to be affected, fundamentally, classically, one must study intracellular potential of the neuron, and that too in a behaving animal after loss of REM sleep and otherwise in a normal, which technically is difficult to study. Even today, what to talk about 30 years back. Therefore, what I thought, REM sleep loss would affect factors modulating neuronal excitability, and that all of us know that neurons have sodium-potassium ATPase, which is maintaining the transmembrane potential. So I thought the hypothesis was REM sleep would maintain brain excitability, and in order to maintain that, it has to affect sodium-potassium ATPase. In order to prove my point, if we have to do it in the animal, first thing, whether the rat has it, and I have to show it in the lab. Of course, other people had shown. Here, this is the record. It does go into REM sleep. So we deprived the animal and took out the brain. And as you can see, these are the first study from this establishment of the lab to study REM sleep research in this country. And I had to study, I had to standardize the whole process in 90s, late 80s and early 90s. And this was the first paper in 93 to show, yes, the brain sodium-potassium ATPase activity is increasing by about 60, 65, 70%. After deprivation, we had all sorts of control. Then we went on. Why? The question is, why should it and how should REM sleep deprivation induce such effects? In the sense, REM sleep loss is a behavior. If I do not allow somebody not to go into REM sleep, why should it affect the biochemical or the chemical processes? So I hypothesize the REM sleep disturbance then must affect the neural circuitry which is modulating REM sleep. And there must be some factor which is getting modulated because of the circuitry disorder, which will be affecting integrity of the neuron, be it in the short term or be it in the long term, and that would affect. This was the hypothesis. The question is, what should be the factor? Then we needed to know the function, how the REM sleep is regulated. So that is another line of work which I will not talk about, but to bring home, to make this lecture go smoothly, before I started mid-80s, late 70s, there was this hypothesis that two types of neuron. One, this is black REM off neuron, which cease firing during REM sleep. And this is a cholinergic neuron. This is known as REM on neuron. And people had proposed that they are connected like this. So acetylcholine inhibits noradrenergic neurons, and therefore REM sleep comes. To cut the story short, we worked on it and showed it is not like that. There is a GABA interneuron, which I will not go into detail. And then we worked out how these connectivities are present. Then we worked out different receptors, subtypes, neurotransmitters, how they are connected, and how the wakefulness area is modulating the REM sleep related areas. And ultimately, we worked out this mechanism, this neural circuitry. The essence here is, these noradrenergic neurons in the locus ceruleus that stopped firing during REM sleep. In the sense, these are the REM off neurons. Here you can see in a behaving animal, these stop firing during REM sleep. Means what? If a neuron is active, as you can see here, it releases neurotransmitter. So if during REM sleep, these neurons stop firing, that means the brain is withdrawing that neurotransmitter. And these neurons are located in locus ceruleus, as I am showing here, and that has the maximum projection all over the brain. And this locus ceruleus neurons are responsible for maximum noradrenergic system into the brain. So normally during REM sleep, these neurotransmitters are withdrawn. However, they continue firing during REM sleep loss. That means what? The brain is flooded with noradrenaline. If that is there, that means, as the last here I have written, REM sleep loss does not allow REM off neurons to cease activity, resulting in elevating noradrenaline level in the brain. So my hypothesis was, this noradrenaline should be more, and that should be causing the REM sleep loss associated symptoms, be it sodium potassium ATPase or any other thing. So how to prove it? The first point is whether noradrenaline actually indeed increases. Here you can see the red continuous line. Indeed, after deprivation, noradrenergic neuron is increasing all over the brain, be it in frontal lobe, cortex, dorsal raphe, PPT, LC, except in hippocampus, which I will not talk about at this moment. By and large, in the brain, noradrenaline is increasing. So that is one of the hypotheses that I made. Now, what happens? Does this noradrenaline responsible for ATPase activity increase? This slide is very complex. Is a lot. I had made it into one slide, almost 3/4 of a thesis data. But to prove my point, please concentrate only on the left first one. All are rest similar. Here, the left 4 bars are free moving, the green one, then black one. After free moving, what we are giving a beta receptor blocker, adrenoceptor blocker. The red one is alpha adrenoceptor blocker, and the magenta one is alpha 2 receptor agonist. The idea is, if we give this blocker, then if the effect is because of noradrenaline, that should not happen. And difference between postsynaptic receptor like alpha-1 and alpha-2. Alpha-1 is acting on the postsynaptic receptor and does not allow noradrenaline to act. Whereas alpha-2 is presynaptic and will not allow release of noradrenaline receptor. If that be so, and the right 4 bars are after deprivation, which is shown here and the lower this thing. So, and then we have normalized the data. I have shown you that after deprivation, there is 70% increase in ATPase. So green and the light blue color, light blue certainly increased, but to make my point, I have normalized to make it 100%. There, if you give beta blocker, like black one, be it in normal or this violet color, there is no significant change in ATPase. But be it red one or orange one, be it in normal or deprived, see, they are bringing down ATPase activity to 60 to 70% level. But if you give alpha-2 agonist, that means you are not allowing noradrenaline to be released, then it is coming down to almost very low, almost 10%. Means what? ATPase activity is maintained, but the elevation after deprivation is likely to be these noradrenergic system, which is supporting my hypothesis. And mind it, this is in mid-90s study. Then the question, how is it working? We worked out the intracellular mechanism. This is in 2000, how through phospholipase C and then phosphorylation of this and how calcium activation. But then here there is a dephosphorylating step, calcineurin and acting of this thing. Here the question mark, as you can see, because in this study we did not do, but that was, I had shown this arrow because that was in the study literature, that ATPase activity dephosphorylated form is an active form. Subsequently, in 2007 or 8, we did show that is happening, but in the interest of time, I'm not showing that, but I'm showing the later on. This is the entire intracellular mechanism that we worked out, that how elevated noradrenaline after deprivation, on one hand, by acting on alpha-1, is closing the calcium channel, and on the other hand, by phospholipase C activation, then it is getting released 2 of these calcium ions, which is there on the ATPase molecule, and then activating it. And thereby, this ATPase activity is increasing. The question here then, if this is happening, this can happen in a short term. How will it affect in the long term? In order to have sustained effects in biology, it needs a transcriptional change as well as epigenetic regulation, both at the noradrenaline level and ATPase. And then if we talk about behavior, then it has to affect at the hardwiring this neuronal synaptogenesis, which ultimately will affect the pathophysiology. So if it is there, how does noradrenaline get synthesized? Here it is through DBH and PA genes, and there the transcription and then transcription of the enzyme, which is tyrosine hydroxylase, which ultimately will induce noradrenaline. And so we worked on it. And as you can see for tyrosine, DBH, dopamine beta hydroxylase or tyrosine hydroxylase, we worked on how the methylation or acetylation, how they are modulating, and we have worked on the epigenetic mechanism as well, which justifies that noradrenaline level is increasing in the long term. Then what happens on ATPase? And as we know, ATPase is of tetramer, alpha-1 and alpha-2, and there are, of course, alpha-3 as well, and then beta-1, beta-2. What we found, and indeed that be it alpha-1, be it alpha-2, be it alpha-3, all these are increasing after deprivation. Then does it happen because of noradrenaline? Yes, indeed. Because if in the deprived one, if we are blocking with prazosin alpha-1 blocker, it see that alpha-1 is coming down, which had increased, as you can see alpha-2, as you can see alpha-3. Now that means the transcription is increasing of alpha subunit, which is the regulatory unit for the ATPase. So the transcription has to work in this way. Therefore, we wanted to study how the transcription is increasing. Look and behold, see the difference. mRNA transcript is not increasing any of them. But we saw the transcription has increased in the Western blot. So we thought there must be some changes in the translational, post-translational modification. How will it happen? Then we worked on by stabilizing, then probably RNA stability study has to be done. The RNA stabilization is increasing. This part we are preparing now for submission. We have worked on it. See, noradrenaline is stabilizing. Otherwise, we are doing with actinomycin D. The time is not that much to explain all the mechanism, but just to say, the pink one, what is this color? Yellow color. Normally, if we stop RNA synthesis by actinomycin in Neuro 2A cell line, then it is degrading like this. But if I give noradrenaline, the degradation is lost, be it for alpha 1, be it alpha 3, be it alpha 2. Means what? It is not allowing the degradation. Now, how does the mRNA degradation take place here? Normally, it is known that mRNA is coming out of the nucleus, and then here the transcription is going and translation will take place. And once that mRNA has translated into protein of ATPase alpha 1, alpha 2, alpha 3, there is an exosome which is preventing or breaking down the mRNA so it doesn't go into cyclicity. What we are finding, noradrenaline has a role where this HUR is a transcription factor, which it is getting released and increasing here, and thereby it is not acting the exosome to act on it. The same mRNA is repeatedly synthesizing more of ATPase. Therefore, it is stabilizing this noradrenaline and increasing ATPase activity. And that can be seen here, how HUR is coming out from nucleus to cytoplasm. This we have done Western, and here we have showing how these are happening. And these are transcription, as we showed the transcription by RT-PCR, how things are happening. Okay, now coming back to this, I talked about ATPase and therefore the behavioral changes. Now at about 15 minutes that I have at my disposal at this moment, let us talk about hardwiring. What is this hardwiring? As we know, the brain, there are synapses. And as we know, the neurons grow and make synapse. And that synapse can change for the ultimate permanent memory. That means if REM sleep is affecting memory, REM sleep induces memory for long-term memory synthesis, or deprivation is affecting, then I should be able to show that this noradrenaline will have effect on the synaptogenesis or withdrawal or breaking down of synapse. Then we standardized neuron to trace of about 250 to 300 micrometer thick section, and then trace a neuron, as all of you can see, the branching and the dot where the synapse means spine densities that you can see. So here you can see the nodes where the branching takes place. This is soma. These are the axons and then dendrites. And all these on the top, these are the spines where the synapse is taking place. And here is the behavior of the rat that you are showing. That means if deprived rat, it is taking long time to reach the platform. Whereas here, just one go it has reached after learning. This rat has forgotten. And this rat, if I take out the brain, see the branching is much more broken as compared to this branching here, which will be more clear in the next. Yeah, here you can see. Here this is in a control one, how much branching in the hippocampal neurons. In control, there are a few have broken, I understand. But after deprivation, see, these are much more broken down. So now the question is, how is it happening? Does it happen with noradrenaline? Oh, this is a confocal microscopic picture to show I think it is little hazy, but at least you can see the green one is tubulin for neuronal structure, and the blue one is for Hess staining for nucleus. After deprivation, you can see the nucleus is disintegrating and the neurons are disintegrated. Means what? After deprivation, the neurons are breaking down, which justifies what I showed you here, that the branches are breaking down here. Now the question is whether it is happening with noradrenaline. Well, we did this slide is showing the ultrastructure with TEM to show that there is a blebbing, and because of that, apoptosis is taking place. You can see this is in 2006 publication, and this is 2016, little better type of blebbing, and the chromatolysis is taking place of the nucleus, and therefore apoptosis is taking place of neurons. Now, does it happen with noradrenaline? As you can see that, yes, it does get little reduced in prazosin treated. Means not, if you can see here, REM sleep deprivation as compared to control chromatolysis, but with prazosin alpha blocker, the chromatolysis has been reduced. Therefore, it suggests that noradrenaline certainly is trying to prevent it. And here you can see, be it bad, which is increasing, all the factors for apoptosis, be it bad, be it APAF1, be it caspase 3, be it cytochrome C, be it caspase 9, are increasing while PK80 is decreasing. Significantly, caspase 8 did not change. What does it indicate? Indicates with the general classical knowledge that extrinsic factor is not being affected. The intrinsic factor, the mitochondria, is getting affected, and we worked out that this is the mechanism through increased noradrenaline, how the entire pathway is happening, and therefore neuron is getting into damage mode, neuron is getting lost. But how does it happen? I'm showing it in an animal that this is happening. But how do I prove that this is because of this noradrenaline? As we know, the neurons have multiple inputs. So what the idea is, if this black neuron is like locus coeruleus neurons, which is projecting all over the brain, and if this noradrenaline is causing the neuronal damage, idea is if I can inhibit the synthesis of noradrenaline and then deprive the animal, then I should not see this ATPase activity increase, which I showed you earlier, and I should not get the apoptotic markers. How do I work on it? We have worked it this way. In the same animal, this is control. On one side, we are giving shRNA. On the other side of the same rat, we are injecting, I am showing it here, we have given control shRNA. We have also done for siRNA, as you can see the Western blot here. I am showing representative picture. In this animal, if I have given shRNA and waited for about a week, and when we have done siRNA, we have waited for 24 hours. What you can see here, the number of cells, shRNA of TH, tyrosine hydroxylase. So if tyrosine hydroxylase is not synthesized, then antibody of TH will not show. That is what you see here on the left side of the same animal, mind it. So on one side we have done this, on the other side we have given control, and we have then counted the number of cells. In a normal count, if we are saying in an area, here is about 450 or odd cells that we could count in that area. After TH, shRNA, there's almost 1/5 here that it is decreasing. And there you can see, even with that Western blot, at least for siRNA, there is significant decrease in TH. Means what? Those cells are not giving noradrenaline. Then in such animal, we are depriving of REM sleep. Look here in this rightmost. If we are giving control siRNA and depriving the animal, ATPase activity is increasing by about 70%. As we had shown in 1993, I showed you this slide. For another study, we have shown in 2003, there is 70% increase. Here also it is increasing. But if we have given siRNA, or for that matter, shRNA, and waited for 7 days, see here, even if we are depriving, there is no ATPase activity increase. Means what? The symptom we will not get, and behaviorally, yes, we do not get that symptom, what we used to get in otherwise normal. Look here, if we are going for cytochrome C, if we are giving control siRNA, which is increasing as I showed you earlier in apoptosis, that is not happening here. That means cells are less damaged. And here tyrosine hydroxylase, which normally control it, is increasing, but if I'm giving siRNA, it is not happening. And here is the Western blot that you can see. Now, what is the meaning here? Earlier, I was saying all the defects or the difficulties or the damage of deprivation is because of noradrenaline and it is creating the problem. And here I am saying the noradrenaline is beneficial. And if it is a damaging effect, why in evolution nature has retained noradrenaline? So that was the question. So to cut the story short and go it little swift, one of the things that we have to understand, brain is highly metabolically active, but it has the least antioxidants. That is what you can see here. That is what you can see here. Brain has least level of antioxidants as compared to heart, liver, kidney. At the same time, brain has high level of iron, which favors oxidative stress. So can that be linked? And then my question was, can it be linked with noradrenaline? Then we went into 1960s old literature and we found noradrenaline has an antioxidant effect. So can that be linked? What we found indeed, in a deprived animal, see, ROS generation here, this is left panel is in deprived animal taking out the brain. After deprivation, ROS level and lipid peroxidation level is going down. But that is not there if we are treating with prazosin or clonidine, which is not allowing noradrenaline to be released. Then we did in vitro, means we are taking out synaptosome and did it. Noradrenaline indeed reduced ROS, indeed reduced lipid peroxidation. So if that be, this is ROS and upper one is lipid peroxidation. Then we did in the cell line to understand the mechanism. If we induced H2O2 induced ROS generation in Neuro 2A cell line, see, by noradrenaline, it is reduced. That means noradrenaline has a preventing mechanism. It is so complex when we saw these our results. Then we found a significant number of sleep loss related disorder is iron related. And there is a Fenton reaction that ferrous can go into ferric ion because of this oxidative, and then hydroxylation oxygen is generated, ROS is generated, then that goes into cell death. In a normal case, not in brain, SOD and glutathione, because of the antioxidants, this is reduced. But in case of brain, it is not there. Wait a minute, if it is not there, what has nature created for such a complex thing? Then we found, indeed, noradrenaline is chelating ferrous and not allowing it to go to ferric and preventing it to go there. And if that is there, because I may have only 2-3 minutes, what we did, we tried to understand that in primary culture. There also, we developed this method to do it from the egg, taking out the brain so that we don't kill rats or pups or others. Then this is what you can see here, how the neurons in a culture form synapse. Then we went ahead and tried to understand how the neuronal branching is affected by low and high dose of noradrenaline. Here you will find lower dose of noradrenaline is increasing the neuronal branching, where higher dose 50 and 100, it is damaging the neurons. This is here just a picture of here, and this is antibody stain, where you can see the branching is increasing in lower dose, but higher dose the neurons are damaged because you do not get the green fluorescence and the nuclear fluorescence. The cells are damaged. Means what? This is what these neurons of REM of neurons, which normally is stopping. That means the brain is washed off noradrenaline. So as you have seen, low dose of noradrenaline is helping neuronal growth and branching and synaptogenesis. Therefore, during REM sleep, when noradrenaline is withdrawn, we know that REM sleep is helping memory formation by forming the branches and synaptogenesis. However, if REM sleep loss is there, the neurons get damaged. And this neuron locus coeruleus gets input, be it from preoptic area, dorsal raphe, prepositors, hippocampus, substantia nigra, be it all different forms of hormones. And as I said, the whole brain gets 80 to 85, 90% noradrenaline from these neurons. And they get affected in this way. So in nature, what it has created, normally noradrenaline is protecting the brain because REM off neurons are on. However, when this noradrenaline level is going high, then REM sleep is coming so that it is stopping these neurons, so the brain is withdrawn of noradrenaline by washing off, and it is helping neuronal or the brain protection. However, if REM sleep deprivation is going on, those neurons are continuously firing, and this noradrenaline protection mechanism, even if it is there, the antioxidant effect otherwise is getting lost, and it is affecting other cascading effect because of higher noradrenaline acting on other subtypes of receptors, and therefore it is creating damaging effect on the neurons. And therefore, even if what I have written, noradrenaline acts as antioxidant to protect neurons by preventing Fenton reaction. Low dose protects, while high dose causes damage to the neurons. And this is what ultimately we have shown, and my hypothesis is, and which I have proposed in 2011, the REM sleep maintains as a housekeeper of the brain and maintains the neuronal integrity of that. So though one spends the least time in this cognitive states, through evolution, REM sleep has evolved, and it is maintaining this noradrenergic level so that it reaps, is a highly efficient process so that we can maintain our integrity of thought process and cognitive processes. Therefore, evolutionary, it is still maintained even for millions of years. Therefore, I have written this as an editorial: never sleep on sleep and don't lose sleep on lost sleep. Why sleep when sleepy, lest you become sleepery? With this, I will end my talk, and it's my pleasant duty to acknowledge this slide is for all my students who have done left side of the done PhD. These are the people who are at this moment with me. There are postdocs who here, like Rachna and Bindu. They are again at respective places. Other people who are here, they were either in project or something. There are other people also who are in projects. But here I have all the names who has been there in some publication. There are many, many people who spent some few months to some time in the lab. But since no publication has come, otherwise there will be hundreds of students' names. Like I have seen, many people are in US or Canada or many other places. So I'm not naming each of them, but everybody has contributed some form or other. And the funding has come from all taxpayers' money, as it is shown here. Ultimate and finally, and not the least, I am grateful to all the animals who sacrifice their life for us to collect this information. And I hope this benefit to the human being. Yes, some of these things do benefit some human being. The clinical people do use them in some form or other. With this, I stop here. Thank you very much, everybody.
Thank you very much, sir, for your wonderful talk. We have been interested a lot from your presentation. And there are some questions in the chat box. I am requesting Dr. Raghavendra Mishra to present them to sir.
Possibility of abnormal firing, yes, that is always there. I could, what is the, yes, it is always there. That's the question. I think the person has to ask me again. Abnormal possibility means what? Oh, see, I have primarily, then would you give me time? I have to bring back, okay, let me see. Let me see. See, I would suggest you go for progress in neurobiology. Uh, yes, this cannot be said in a few words. I will just show it to you, that slide. If you see this, you can see this is a simplistic picture. Here, this is noradrenergic neuron. Here, these are cholinergic neuron, then GABA. Then there are other neurons. Here, there is orexinergic neuron, TMN, there is histaminergic neurons. So there is a balance. But essential part is these noradrenergic neurons must stop firing for REM sleep. Now, how do you prove it? We have proven biologically by stimulating these neurons electrically, by giving GABA blocker, by giving agonist of GABA, by cholinergic, by noradrenergic. We have done in many ways. And not only that, or orexinergic as well, not only that, then we have done mathematical modeling of the entire circuitry. And what we have shown that if these neurons are kept active by the wake active areas, irrespective of whatever you do, REM sleep will not come. This is extremely difficult to do biologically. Therefore, you do need physical or mathematical modeling. Computer simulation is a great advantage these days, and this we have done in 2012 because I had a physics student who was working with me. And then we worked on it. So there is a balance of all different neurotransmitters, and there will be disturbance, and disturbance in any one of them might cause a affect REM sleep. But ultimately, it has to be noradrenergic neurons which has to stop. Therefore, to tell you in one word, it will be extremely difficult because this is a very complex. Certainly, noradrenaline is helpful, but there will be many supporting and allied neurotransmitters, which many of them will be affecting to cause damage or disturbance in sleep or REM sleep. So if you are interested, I would suggest you please go through this. This is almost a huge review, a big review here, and you will get some information from there. Okay, thanks. See, what I would suggest, so far we know of, hippocampus is doing these memory. Now then, I am not going to take a memory class at this moment, but let us take it forward from here. And this, if you give me 1 minute, the paper which is under revision now, and we are trying to send the revised version, there it will be much more clearer to you. Give me this, I will bring back those slides, which is not here, to show you exact picture what is happening in hippocampus, those pictures. See here, I showed you these branches, and here is hippocampus. You can see here, this is CA1 area. If this is normal neuron, see the branchings are broken down. And here is soma area perimeter is not significantly changing, but you see here the length, the basal apical dendrite length, the basal dendrite length. More importantly, see the nodes. I showed you this nodes right here. If you see here, these are nodes where from they are branching, and here these are spine number. So it takes almost 7 to 8 hours to trace one neuron of 250 micrometer thick, and then carefully to count each of these nodes. As you can see here, how the apical dendrite nodes are significantly damaged after deprivation here. Basal dendrite, see, significantly damaged. The spine densities are reduced and the neurons are breaking down. This is here CA1 area and here it is CA3 area. See the branching, how they are significantly reduced here. And then here the spine, we went into how the stubby spine, how they are breaking down, and these are the numbers that you can see here. This is a mushroom spine, which is the more a permanent memory, increases the mushroom spine, which is significantly decreased after deprivation. And here, for temporary effect, you can see the synapse ultrastructure in TEM. See the length of these, length of active zone is significantly increasing to show as a compensatory phenomenon. So if the length is increasing, so receptor number will increase. If the receptor number will increase, even if their branching is reduced, they are trying to compensate, but it is not affecting. And here, as I am showing, then you are seeing MUNC18, which and then postsynaptic density protein PSD 95. They are trying to increase for a compensation, but if it is going on for a long time, they are not changing, and all these are happening with noradrenaline. This is what I was showing, how they are breaking down. Now, in short, even if it is time is short, did I make sense, and was the question or the answer clear to the questioner? See, the point is, this will all, this is summarized in my, this will be summarized in my, this slide, which I gave you here. Can you see my slide here? See, noradrenergic neurons are the locus ceruleus. And I said it is the area which gives 80 to 90% noradrenaline all over the brain. This does have an effect. You think of any area in the brain, preoptic area, thermoregulatory area, hippocampus, which is going. It is going from SNRPR, means substantia nigra. It is from amygdala. It is from orexin. It is from dorsal raphe. It does get effect from other brain areas, including reticular formation. Therefore, if you keep this active by any input, it certainly will be getting affected. But then, as I said, it has to be a balance, different inputs. If you, I do not have time, otherwise I would have shown how the reticle, we have worked on how these locus ceruleus neurons get input from, be it rostral brainstem reticular formation, which induces wakefulness, versus preoptic area, versus caudal brainstem reticular formation, which induces non-REM sleep. And here these neurons are activated by the wake area and inhibited by the sleep area. But then if you keep in active by some other input, obviously this will get affected. And if noradrenaline release is more and the washing away is less, obviously there will be disturbed sleep. That is what I would expect happens in REM sleep disorder, restless leg syndrome, be it in Alzheimer's, be it in depression, in many of these things. The difficulty is we are getting all these symptoms so complex. The patient is coming to the clinical person or the doctor much later when the disease has already become very complex. Then it becomes very pinpoint to do it. Did it get some answer clear? There was some, though I could make out the question. Can you repeat? Because there was a cracking of voice. You repeat, your voice got cracked. Is it, no, no, listen to me. Is it that the question is, what is the advantage and disadvantage of wired recording versus wireless? See, if we talk about acceptability, what is acceptability? We accept certain things as long as you do not have anything to rebut or anything negative about it. For wired one, the noise will be little less or more depending on what you are differentially eliminating the noise. So when you are doing wired recording, you are coating the wire with less noise chemical, and then you are putting like a Faraday cage, you are wiring. But if you can use it non-wired, means remote recording, one advantage there is where the person can move around. But then because of movement artifact, there may be many other artifacts. But then if these days the technology has improved so much, because of carrier frequency, you can decarrierized and get the signal out. It depends on how best you are getting the signal to noise ratio. So I wouldn't be surprised about the acceptability. If you can show me clear signal by eliminating noise, I will be accepting it. And I guess anybody would do that. And always we have to see what is the resultant pros and cons, how much you are doing it. For example, if you are doing it in an animal, we are restricting it within a cage. But if you are doing it in a wild, then of course cage is not there. Somebody may argue it is a caged animal, it is non-caged animal. For example, in humans, if you are doing it, if somebody is just lying on a bed or sitting on a chair, then somebody may argue you are not allowing the normal behavior. If you are allowing normal behavior, then you have to do wireless. Now, wireless also has a limitation, whether it is a few meters or you are going for hundreds and millions of meters, then you have to develop the technology that way. So present day technology has increased significantly, and then we have to take it. So acceptability would depend on how much you are convincingly recording. So I wouldn't have yes or no straight away answer unless there is a very specific say because of this type of noise which is coming. Well, as you can understand, the question itself is so complex. The thing is, if it is see, for any patient, first thing is needed to give immediate relief. We do research to find out all the effects, that is fine. That does not mean the immediate effect has to be withdrawn. And always the body has a resilience. So steroid treatment, if it is recommended at any point in time, at that point in time to overcome the situation, by all means one has to do. But as the body has a mechanism to get rid of it, to wash it out, we have to do the post-treatment, post-recovery process very well. For example, to give a simple analogy, anesthesia is not very good. But does it mean if we have to do surgery, we don't give anesthesia? We have to evaluate the effect there. So in a serious COVID patient with some complication, if it's recommended to go for steroid, I would think by all means within the limit we have to do. But then one has to be very careful for post-recovery. We have to be careful and give that type of care. If we do that, I am sure it will be taken care of. And the clinician who is supporting should be taken care of in that way. This is something very, very common whenever we talk about sleep loss, damage versus stress. I have just written a review, and this has been historically going on. So now we have done an experiment and we have got reasonable comment. It has come yesterday and we have to submit it after modification, means explanation. But to cut the story short, first thing which I have argued in 2000 in review, and then I have argued in 2000, I think 8, 9, and again last year in review. First thing, we have to define what is stress. The problem lies, see, we are experimental scientists. For that matter, we have to understand what is the difference between philosophy and the science that we do. Scientific explanation, we have to show things very objectively. In that sense, what we mean by stress is doing something which is normally body cells are not exposed to. And that also there is a threshold for it. So many action is stressful, but if one is acclimatized to it, then what is not stressful? For example, can we respire if body was not producing carbon dioxide? So the stimulus comes by carbon dioxide, not by oxygen. In fact, increased oxygen has an oxidative stress, and that is how when the life evolved. It is in a medium when the carbon dioxide was there and the oxygen was a byproduct. Therefore, when we do a biological experiment, is very important, as many control studies we do. Once we do that, any example, even it has been shown by pin pricking during injection to a child becomes stressful. So we are not talking about that. Once we do experiment, be it sleep loss, yes, it will be stressful. But suppose somebody is sleeping at different point in time, but that time is fixed, then it will be less stressful for that person. So in case of stress, yes, if there is increased level of noradrenaline, as I showed, neurons are breaking down. So the memory is lost. And if that is there, there are chances or there are studies that steroid has an effect in growth of neurons. So little bit of steroid is helpful to prevent the stress. Steroids are not damaging as such at initial level. Most of the time it is preventive. But then if it continues longer, then even we do not know the mechanism, but it gets damaged, I am of the opinion that it affects in the secondary way and then it gets damaging. So this is my take, that just by stress doesn't make much sense. If it is acclimatized, then it is not. But we have to take care in the recovery period. Okay, there's a loaded question. Okay, now in order to see, first thing, I am not seeing you people in face to face. Would you give me a minute? Do I bring another slide? It will be easy for me to show. Do you
Yes,
have time?
Sir. Yes, sir.
Okay, then let me go back and try to get a slide from because I was not prepared that this type of thing. Let me bring back a slide. Okay, what I mean by the time I am getting the slide, see do we understand what is lucid dream? We know what is happening, but do we know the biological mechanism? I'm afraid not. So in that sense, but we understand one thing, that somewhere a sort of memory gets activated. Also, one has to understand that if we are dreaming, the dream is not coming throughout the REM sleep. Even if it is coming on REM sleep, it is not that it comes throughout the REM sleep, it comes intermittent. And I think all of us have experienced that the dreams come intermittent. At some time it comes, at some time it does not come. If that is there, my hypothesis is, at certain point of REM sleep, certain circuitry of the brain gets activated and the dreaming is coming. In principle, now that is lucid or non-lucid or damaging or scaring, that comes later, because it depends on which area of the brain gets affected. In that sense, we are working on this. My hypothesis is, once we have this structure of neural circuitry of REM sleep, now I am getting some slides which want to show you to cut the story short, because these are very loaded questions. Okay. See, I have shown like this. Can you see the slide now? Are you able to see my slide?
Sir. Yes, sir. It is visible.
Okay. See, it says, still we do not know why vivid dreams appear. We had shown, and I have shown in this lecture, how REM off neurons and REM on neurons are active. There, I have shown that how this is limbic structure. This is either in the amygdala or amygdala and substantia nigra. These are modulating this circuitry to bring in REM sleep. And here you can see from this, and we have worked out these. One paper has already been published. One is under prepare. We will be submitting in a week or 10 days time. What is there during REM sleep at certain stage? Some of these REM on neurons are active, and part of these REM on neurons are activating limbic structure. If it is a limbic structure for visual, that I am seeing a memory of rose versus I am seeing a goat or a cow or a dog or my mother, that memory gets activated. And why that gets activated? See, this is what is the active circuitry which I have shown. Without going into this, let me tell you now the philosophical angle. My way of thinking is why it comes only during REM sleep. This is at this stage. These are the wave frequency which it comes. If when we are active, the EEG frequencies are very high, during sleep and REM sleep, when the frequency content is going down, there is a resonating frequency. And as you can think of what we have in the middle here, impedance matching, that happens. And when the frequencies from outside may be coming, that probably is, again, mind the word probably. The conscious state is that, and we can perceive this. I had presented this year, February, in Bangalore, when the cognitive science and the Vedic philosophy and Tibetan philosophy. Then we can achieve this type of thought process. And then what is happening? We have generally biological, or the modern science talks about wake, non-REM, and REM. But our Vedic or Upanishadic philosophy, we have Turiya state, which is the fourth state that we know of, which has been talked about. But biologically, we do not have any explanation. My explanation is, and this I have presented or I have spoken in the 3rd chapter of REM sleep book, how our Vedic Upanishadic philosophy helped explaining or give an insight to understand modern science. If you can see here, very mildly written Turiya in between. So what happens during wake? The activity is increasing and goes into non-REM sleep and REM sleep. But if they go in the background, Turiya comes on the top. So probably during this state is coming where this memory or these types of part of the REM sleep, and probably in meditation, if we go further deep into this, we come up this Thuria state, which is in the very low frequency input, which I was referring to here. And therefore, probably in meditation, people can get into this state. And I will not be surprised if this happens in case of our uh, glia may have a role. And in that case, through bioinformatics study, what I have done, see, glia is no glia, no glia, no glia up to porifera, but they have started neuron. So the complexity of thought process and our cognitive things that and the complexity of our thought process, we talk about from here Cinderella or Annelida, that we can say that they have their thought process and can decide. So this way, I am thinking, or my way of thinking, the glia gives the background which I was talking to you here, very low intensity thought process here. Over and above this type of waking non-REM REM sleep is coming, and that is going. In short, masking and unmasking of expressions define our states. Whosoever has asked the question, did it make some sense, or I was very complex? I understand. We need to talk more, I guess. Or I don't know, if there are time, we can more discuss on this.
Thank you very much, sir, for spending your valuable time with us. Thank you once again. Now,
Sir, I
we
have
are
a
to the next part of our session. The last speaker of today's session is Professor Nilkanta Chakraborty.
Okay, thank you very much. Thanks, everybody.
Okay, sir.
Okay, I am going off. Fine.
ma'am, I have a question.
Okay, now we are moving to the next part of our session. The last speaker of today's session is Professor Nilkanta Chakraborty, Professor and Head of the Department of Physiology, University of Calcutta. Once again, I am requesting Dr. Rupali Sarkar to introduce Professor Chakraborty. Please, ma'am.
Here we come to the end of our today's technical session. Our last presenter, Professor Nilkanta Chakraborty, a professor and head of the Department of Physiology, University of Calcutta. And he chose a very interesting topic from our recent lifestyle: anxiety, the good, the bad, and the ugly. Professor Chakraborty, who was the former director of SN Pradhan Center of Neurosciences, University of Calcutta, is a very dynamic and versatile man with a mind of scientific approach. He has been specialized himself in the field of brain and cognitive physiology, neuroendocrinology, and reproductive physiology after getting his master's, MPhil, and PhD from the University of Calcutta. He did his postdoctorate from Morehouse School of Medicine, Atlanta, Georgia, USA, in the field of neuroendocrine versus peripheral peptides. So far, as we concerned, Dr. Chakraborty has published his extensive work in neuroscience, at least in 18 international and national journals like Neuroscience, Neuroendocrinology, Neuroinflammations, Neurochemistry International, Indian Journal of Experimental Biology, and so on. He has delivered at least 25 national and international invited lectures throughout the last 9 years. With such aptitude in the field of neuroscience research, he never hesitated to take the responsibility to guide the students for their PhD degree and to act as a coordinator in different programs of academic staff college of the University of Calcutta. In the year 2017, he also organized with great success an international conference on neurodegenerative diseases at Kolkata in collaboration with INK Kolkata and its Institute of Neurosciences, UAK. Well, this is in a nutshell. Now I invite Professor Chakraborty for his presentation. Professor Chakraborty.
Am I audible? Hello?
Yeah,
Am I audible? Okay.
audible.
Thank you very much. প্রথমেই সবাইকে শুভ ময়লা, সবাই ভালো থাকুন, সুস্থ থাকুন। I'm really thankful to Dr. Pradeep Kumar Maiti, the principal of Arandamon College, and Dr. Joyita Banerjee, Dr. Arindam Dalal for giving me opportunity to discuss, to share some views on the topic that I like to discuss with you, the anxiety. It is really, it is after so much enlightening the talk delivered by Professor Birendran Mollik. It is I do not want to put you so much anxiety. In fact, here in my talk, that includes some psychological events of the anxiety and recent neurophysiological, particularly brain imaging studies in support of that, to find the gradient of the anxiety. And I have included one publication that we have published and we have done in our laboratory, that how anxiety is associated with the memory, to find the gap between the anxiety and its neurophysiological aspect. So first of all, I'd like to just mention the psychological gradation of the anxiety, that is good anxiety, bad anxiety, ugly anxiety. In our daily life, we always are exposed to the anxiety condition. Whatever we do, the anxiety creates in our mind. Now, question is, what is the level? If the level is good enough to get our performance maximally, that is good anxiety. If the level of anxiety is more than that, then you may call it the bad anxiety, or even more, there is ugly anxiety. So that happens when you are in conscious arousal condition. So what is good anxiety? Whenever we in working condition, when we are trying to do something, our brain networks actually create such kind of behavioral aspect which creates anxiety, the behavioral output, your feelings. If you have a positive mindedness, if your brain network emotionally accept the fact, then it is good anxiety. That means you are feeling the anxiety. You are thinking and analyzing to minimize the error, to reach to the top, to solve your performance, the work that you are trying to do. Now, if with the more excitement or over arousal condition, if brain networks induces emotional avoidance, I'm talking about the network, I'll go to that how the brain network actually causes either acceptance or avoidance. The brain imaging studies has been found. Now that level of anxiety, that is bad anxiety, can create, in that condition, you may not get success. With the further adverse level of, more level of anxiety, the irritability or depression may occur. In that case, the sleep disorder is associated with the anxiety level or anxiety-like disorder. That is ugly anxiety. So we may say that the good anxiety and bad anxiety is such kind of physiological level of having some threshold of anxiety, and ugly anxiety is a pathological condition where the anxiety disorder happens. So many researchers going on to rehabilitate the situation, the brain network, the physiological condition, to get it in normal by using the cognitive or the behavioral rehabilitation to uh, go from ugly stage of anxiety to the bad and finally good condition. So in the next slide, here I have given the area of the brain associated with the activation of this area associated with either emotional acceptance or avoidance. You see the area is different. In acceptance, emotional acceptance, the activation pattern is paracingulate cortical area, in the little deeper. Whereas in avoiding condition, the ventrolateral prefrontal cortex, insula is also associated with the emotional acceptance. So this is just a view that there is a different brain activation pattern for acceptance and avoidance. Now, brain will direct you whether with your biasness, because the biasness is very important here in psychologically. Because if you have a positive biasness, that may create a disturbance of such kind of emotional acceptance, or may overlay on that to disturb the normal behavioral condition. On the other hand, negative biasness also can create impact. So we'll go to that point. Another psychological explanation is that there are almost 8 states of the mind. Here, anxiety, worry, apathy, boredness, relaxation, control, ataxia, arousal, all this that has been categorized psychologically. And it is well established for a long time, in 1950s around, that if this emotional acceptance takes part into the brain and provides the brain network and direct you to accept the thing, to feel the anxiety for your performance. So in that case, you will be the happy man, because you will gain and you will perform your work. Recently, it is published in another psychological study that the people who are in anxiety state, high anxiety state, you see this line, this shaded line, can perform better with a certain limit of anxiety than those who have, this is working memory cognitive performance, WMC, which represents that which have a certain level of anxiety, they can perform better if they have their tendency to accept. Okay, and the best example is that the little bit anxiety, the students, or even whenever we do something, the students they have a exam anxiety. Okay, so before examination, every student feel that. Who feels that anxiety? And if it remains in a particular state or level, it is not actually defined what is the level, what is the biomarker of physiological biomarker of this that level of good anxiety or bad anxiety. That's fine. That is the challenge to the neuroscientist. Now, students, if they have anxiety, they have anxiety. If they bear it and accept it, they feel it, then in that case they should have the better performance. It has been found. Okay, and it is psychologically accepted. Then those who do not have any anxiety, it is not possible to have any anxiety before examination. This is the study I have found recently in Lancet Psychiatry. They have done a meta-analysis from a huge study of data bank. They have categorized the psychological state with the different brain network of 6 categories: default mode, silent structure, negative effect, positive effect, attention, and cognitive control. And you see the areas are different in these 6 psychological conditions. But however, certain regions, prefrontal cortex and cingulate cortex, all these are involved. Most of the cases, prefrontal cortex in different position, either dorsolateral or medial, medial. So whatever may be, they have categorized based on the network, the 6 stages of mental status, and where the different regions of the brain are interacting. On that, using that particular state of mental status, they have found the activation pattern and the network status in different anxiety altered condition. So they have categorized 8 anxiety altered condition, and they have reported that how these 6 category of mind state they are imposing on such kind of psychological altered mental condition. Very interestingly, you see the default mode remains in the conscious thought. It is in a resting condition. It's fine. It is almost same in the default mode. When you go to the other condition like the avoidance, see the salience network is coming, but the difference is that the dotted line and the solid line is the hyperactivity and low activity status. So network of the salient network is changing with the activation pattern interaction between these different areas. If you go to the other 2 state of altered condition, negative biasness or the threat dysregulation, here you see the network associated with the negative effect. They are interacting with different mode of network, having little hyper activity between the nodes. Okay. If you go to the other 2, the loss of interest or the context insensitivity, you see the positive effect have different impact. The circle size actually indicates the activity status. The circle having larger diameter have more activity, small one the maximum activity, and the dotted line indicates the less interaction, and the solid line indicates the more interaction. You see, in these 2 altered conditions, the positive effect network, the pattern is different. Similarly, in attention condition or the cognitive dyscontrol condition, you see the network interaction is different here in attention, and in cognitive dyscontrol, if you compare with this cognitive control condition, you see it is an incorporation of default mode, attention mode, and the specific control mode, the region associated of these 3 mental states with different activation pattern and interaction level. So it indicates that the brain networks have specific, there is a specific network for different psychological aspect of mental state or anxiety state in physiological condition or in pathological altered condition. Here that is very interesting that the positive, I have already mentioned, the biasness that actually impacts, it may create the positive impact or the negative impact. That depends on whether it will create the avoidance the network or the acceptance networks. Okay, so what kind of network is generating the brain? In positive biasness, the area is associated with the medial prefrontal cortex, and in negative biasness, you see the cingulate cortex is associated with the negative biasness. Okay, here it is the complicated network with the amygdala, the area which is associated with the emotion, with the hippocampus, prefrontal cortex, and other areas of the brain that indicates, and the different excitatory inhibitory modulation that it is published in Nature Review Neuroscience 2015 that how the amygdala, the area which is associated with the emotion, and the hippocampus which is associated with the memory. You have learned, you have listened that how the hippocampus is associated with the memory formation, and beautiful explanation by Professor Vidyanand Molli. And here, one important thing I just want to mention, that there is a connection with the amygdala with hypothalamus, and that is the point. When we are in anxiety state, our cardiovascular system alters, changes in blood pressure, changes in heart rate. So how it happens, I'll go to that point of explanation. Other point is the cerebellum. They have also connection with the frontal cortex, and obviously other part of the brain through thalamus and in the midbrain to the frontal cortex. In the area, you see, you can remember I have mentioned earlier this cingulate cortex, which is associated with the negative biasness, and the dorsolateral prefrontal cortex that is associated with different modes of the anxiety related behavior. Cerebellum is also associated with the cognitive regulation of anxiety. So amygdala is the center, hippocampus is the another part where memory is forming, cerebellum is another part which is interacting with the cognitive performance and controlling the anxiety state. Here it is beautifully explained that how the amygdala, the emotion created within the amygdala, is interacting through hypothalamus, causing the cardiovascular changes through sympathetic activation. And the other way, when anxiety is creating, the parasympathetic activation actually causes inhibition or irritability or other things like freezing, restriction of the movement, respiratory distress, so urination, defecation, tachycardia. So there are so many physiological responses happens due to creation of the anxiety of emotional alteration here that actually interacts with the hippocampus using the memory and interacting the hypothalamus, other part of brain, to creating the impact peripherally in the physiological system. So that is the explanation, neurophysiological explanation of anxiety, how the brain networks that is happening. If it is positive or you are accepting the anxiety, the level is good, then you will perform better. Here I would like to mention this very interesting thing, that two steroid. You know, I mean, it is well versed that we are very much well versed that when hypothalamus is activated by anxiety level of the amygdala, it causes the elevation of cortisol level. That is very common thing, that elevation of cortisol in the anxiety. Now question is whether the brain cortisol level is good for the brain function, the anxiety. Actually, cortisol have a dual role. In certain concentration, it helps the synaptic transmission or the synaptic plasticity. It is experimentally proved. In other condition, which is very well versed and well reported, that the high level of cortisol interacts with the glutamate NMDA receptor, causes exocytotoxic effect through entry of calcium to the postsynaptic neuron, causing damage of the neuron. And that is well reported fact. But the new thing is that the cortisol can enhance the memory and control the attention. I'd like to mention the very recent publication in Neuropsychopharmacology in 2020 by Siddhartha Datta, who is doing now postdoc in NCBS under Professor Sumanta Chatterjee, working on the anxiety, and they have published the how cortisol level in acute stress condition, the certain level of cortisol can causes the synaptic plasticity, and it can help the memory formation. Siddhartha did post PhD in my lab under my supervision, and here you see they have explained beautifully that how the amygdala area, in the amygdala area, the synaptic plasticity is changing. So I'm going to the next part, the how Siddhartha did PhD, where when he did his PhD in PhD tenure, he did a very preliminary work in his under his PhD thesis, that the focus of our study was to correlate the anxiety with the memory. Is there a relation of the anxiety with the memory, and how it differs, or is there a neurophysiological aspect? So our motto was to find that what is the gender effect, and in case of the whether gender effect, and also I mean the difference between the male and female, and also the reproductive cycle, to establish the fact that the reproductive hormone, that is estrogen, whether it is involved in anxiety memory interaction or not. Okay, so what we did, we exposed the it is published in Physiology and Behavior in 2016. So we exposed animals, mice, albino mice, in the early stage of night, around 8 to 9 p.m. to the high illumination of white LED light to create anxiety. And we measured the anxiety by elevated plasma. And also we performed some behavioral studies. We did not do any, actually this paper did not include any molecular aspect. We did simply behavioral studies, and we published the behavioral studies. So what we found in behavioral studies, very interesting, is that we did measure the anxiety in APM elevated plasma. We measured also the motor activity in open field test, and memory test, object recognition in open field, using the recognizing the object. So what we found, analyzing the result, that male and diasters female, when we are exposing to the animals to the high intensity light at night for a certain extent for 1 hour, whether it is creating anxiety or not. Very interestingly, in case of male and diasters female, anxiety is not creating, but memory is deteriorating. Whereas in proesters female, the locomotor activity is decreasing, anxiety is naturally, the anxiety actually causes the freezing of the animal. They restrict their movement, but there is no alteration of memory status. So what we concluded is that estrogen might have a role on the anxiety condition and the memory or memory recognition procedure. And there is a correlation. How they are correlated? There is a correlation with anxiety and memory. Okay, we do not have the exact uh, role here, but still it indicates the estrogen might have a role. And it is reported that the estrogen have dual, like cortisol, have dual role. In one way, when it reacts with the beta receptor, it causes anxiolytic behavior. Where? In amygdala and hippocampus. Mind it, in hippocampus. Amygdala, area associated with, excuse me, I had a call, I have to decline it. Okay, interruption. You just think, you can remind that the amygdala, the area associated with the emotional performance, and the hippocampus area, the area associated with the memory formation. Where if estrogen level, the level is very important, like cortisol also. If it, and also the level of the what kind of receptor is there. If it is beta receptor, then it will cause the anxiolytic behavior. And if the receptor is different, G protein receptor 30 in other areas like paraventricular nucleus, septal complex, or the supraobjective nucleus, through signaling pathway, IP3 pathway, it causes the angiogenic behavior. So, level of estrogen, type of receptor, which area the receptor is expressing this type of receptors of estrogen is very important. So finally, the conclusion is that of my talk, in very brief way, that the brain network, either emotionally acceptance or avoidance network is creating or not, whether the positive or negative bias network is creating or not, whether the attentional or contextual sense, the acceptance of the conceptual thing, whether you are feeling the sense that that particular network is creating or not, that depends, actually, that guides your brain to have a good anxiety or bad anxiety. And in addition of that network, the brain estrogen level and cortisol level can act, but still there is no direct explanation. At least I do not have the evidences that how estrogen, cortisol, and these different kinds of brain network, they interact to each other. But still there are some clues to define that the good anxiety and bad anxiety, they have some physiological aspect. And in near future, the neuroscientists or the psychologists, they can find some neurophysiological biomarkers like the estrogen level, receptor level, network, interaction of these things to define the good or bad status of the anxiety. And obviously, alteration of this level or the biomarker to explain the fact of anxiety disorder. So hope for the best, some new finding in near future. And in this pandemic situation, we are really in anxiety state. We are confined in the home. Although we are interacting over online, that is giving some relief to us. Thanks for that. And thanks for your patience to listen my brief talk. Thank you very much.
Thank you very much, sir, for your nice presentation on anxiety, the good, the bad, and the ugly. Now, it is the time for question answer session. I am requesting Dr. Raghavendra Mishra to deliver the questions to sir.
Okay, please. Thank you. Mhm. Molecular mechanism of creating anxiety in the student, or for those students, those who are not are habituated with the online class mode. Is it? Is it the question? Hello, Raghavendra sir. Okay, use of, that means so that is the online. Hello, I could not get you the point. Will you please repeat? First of all, whether the use first of all the whether the use of the smartphone creating anxiety or not. Okay, fine. Sometimes it is really helps for relaxing you. Okay. However, there are different aspect of the smartphone using which can create the anxiety. That's fine. In fact, long time using of the smartphone, the illumination, the light, the actually the screen. I mean this. Mm, Cell phone screen, they emits they emit a huge blue spectrum light, and that affects that creates a activation in the retina, specialized uh, uh, ganglionic cells, uh, which have a melanopsin, which are very sensitive to the blue spectrum and can alter the melatonin level through retinohypothalamic tract that goes through different I mean 1 2 3 4 neural pathways. There is a pathways between retina to suprachiasmatic nucleus, suprachiasmatic nucleus to paraventricular nucleus. There's a GABAergic neuron which tune the pathway, and then from paraventricular nucleus hypothalamus to pons area through superior cervical ganglia to pineal. Okay, so actually the high lumen what is called cell phone, they causes the alteration of the melatonin level and cause the sleep disorder if long time use at the night time mainly. So that is the cause. One of the cause is that sleep disorder, and sleep disorder is associated with the anxiety, creation of the anxiety. Okay, so that is a one neurophysiological and obviously cellular molecular aspect associated with the anxiety by using cell phone. But other aspect, there are so many things, which is the what kind of activities it is he or she is using the cell phone. Like the children who are using, I cannot remember what is the name of this game, which is now banned. That created lot of anxiety and committed them to suicide. So you see, first of all, what kind of mental status you have, what kind of brain network, either accepting emotional accepting network is creating, or it is creating a positive or negative biasness, any biasness is creating or not. What is the level of the neurotransmitters or different kind of neurotransmitters, mainly the aminergic neurotransmitters, because I have not mentioned that the amygdala, they have a huge GABAergic interneurons which controls the emotion. So it is the inhibitory neurons. So, and there are so many excitatory inhibitory connections with the amygdala with the other parts like hippocampus, hypothalamus, higher brain, frontal cortex. They controls the activity. Now, what kind of mental network is creating to produce your behavioral aspect, that depends. So ultimately, if it does not cause, the use of the cell phone does not cause, any negative biasness or the avoidance, or in that case there will be no anxiety, or you may have a good anxiety even for better performance. So I'm not sure how much clear it is, because it's still a lot of work to. The other people are working the cell phone, that whether it is creating any impact, creating any impact on the physiological aspect and also in the brain. So this is the very basic question of the neurophysiological aspect. Actually, synaptic plasticity, I think you have listened the beautiful explanation by Professor Birendranath Mollik, that in hippocampus, the area of the memory formation, the structure of the synapse is changing. Considering the area of the connection between the two synapses, pre and postsynaptic synapse, and the molecular interactions happening with the pre and postsynaptic terminals. Now, question is, what is synaptic plasticity? There are three states of condition of synaptic level, synaptic structure associated with the memory. What is the three states? Number one, either the synapse will remain and strengthen. Second, either it will remain but remain inactive, or it will be destroyed. There will be no connection at all. Suppose you have 5 connections in an area like hippocampus. In brain, the connection actually happens with the protrusion or the projection that is called spine from the dendrites. So these are the connections, synaptic connections between the spines originating from the dendrites. And that synaptic spine, so you can see like a comb-like structure. If so, there is a connection of 2 neurons having the dendritic connections, and they have a lots of comb-like synapse. And this synapse interacts with the this spine interacts with the other spine of the dendrites of the another neuron. So a billions of spine interaction is there in the brain. So with a particular context, when you are doing something, you are learning first time, or you are using the memory for executing some kind of work within the brain, in hippocampus, in other brain areas which are associated with the memory performance, that is amygdala also part, hippocampus, cerebellum, cortex, prefrontal cortex. There's so many, temporal cortex. Okay, so particularly in hippocampus, it has been found that the spine morphology of the spine changes. Suppose you have 10 connections, either 10 connection will remain, or it will become 8, or it may become 12, whatever may be. That depends on the connection is how many connection is active or not. Suppose for memory number 1, you have 10 connections. For memory number 2, you have also 10 connections. But the array of the order of connection is different for 1 and 2. Like the binary digit of the computer memory formation, like 0, 1, 0, 1, 0, 1. If you consider 0 is inactive, 1 is active. So memory 1 may have combination of 0, 1, 0, 1, 0, 1, 0, 1. In case of memory 2, it may be 1, 1, 0, 0, 1, 1, 0, 0, like that. So that depends on what kind of interaction, spine interaction you have. And billions and billions of spine interactions, alteration of the spine interaction actually helps to formation of different kinds of memory. The question is how the brain retrieving the memory when you are using some kind of working condition or the contextual thing. So here the brain network is very important. Now, within the hippocampus, the synaptic plasticity, that means the morphological pattern of 0101 or whatever it may be, is changing. And the next question is, this change of synaptic plasticity within the hippocampus, whether it is altering the brain network with the hippocampus with the other part of the brain, cortex, the temporal cortex, frontal cortex, amygdala, hypothalamus, cerebellum, whether is there any change of network, then it will create different kind of behavioral aspect. So cognition is nothing, is a process where the sensory input goes, influence your brain, causes synaptic plasticity. Yes, I'm telling this causes a synaptic plasticity because I can tell you one thing. That is a report. I do not have that slide now, but it is a well report, and there are so many reports there that when sensory input goes in hippocampus every day in dental gyrus, one stem cell converts into new neuron and replaces one cortical neuron. So you can just think about that, the regeneration of one, that is neurogenesis, one neuron per day for adult in adult brain, in hippocampus, in dental gyrus, may create the connection, alteration of connections in the cortex. So that is the logic is that when the sensory input is going to the brain in the hippocampus, brain is receiving, it is creating some kind of network and giving the behavioral output. That is your expression. That is the cognitive function. And obviously, the synaptic plasticity is a part of within the brain which is associated with this creation of the brain network to guide you such kind of behavioral aspect. So I'm not sure how much clear to you. Okay, thank you.
Thank you very much, sir, for your nice explanation. Thank you, Raghavendra. Now we are at the end of the session. Thank you to all the eminent speakers of today's session. Through your excellent deliberation, we all have gained knowledge on the recent trends and scientific updates of the neurophysiological processes underlying various brain functions and diseases. We express our sincere gratitude to you all for sparing your valuable time with us and anticipate your cooperation in future for such program. Now, I am requesting Dr. Shanat Kumar Chatterjee, HOD, Department of Physiology at Ananda Mohan College, to conclude today's session. Please, sir. Sir, Shanat sir, please unmute yourself. Okay. Probably sir is not here. Okay. Now, thanking you all of you, the eminent speakers and the participants. We are concluding our today's session. Thank you, all of you.
Thank you.
Thank you, sir.
Thank you, all of you.